Waardenburg syndrome is a genetic disorder in humans. And in this genetic disorder, there is a loss of hearing in addition with some pigmentary abnormalities. And the disorder in the pigmentary abnormalities results in two different eye colors within one individual. So this Waardenburg syndrome was named after a Dutch biologist who noticed that the individuals who have the pigmentary abnormalities also have the hearing problem. They cannot hear properly. So by 1990, many biologists, they perform many experiments to find out the location of this gene that caused the Waardenburg syndrome. And they come up with the conclusion that this gene is located somewhere on the chromosome number 2. But they could not identify the exact location of that gene. Second clue was that some of the scientists, they were working on the mice mutants. So they have also identified in the mice, and they call some of the mutants as the splotch mutants, because in the splotch mutants, the mice also have the same pigmentary abnormalities and have white spots in patches, just like the patches in the human with this Waardenburg syndrome. So the genetic mapping was then conducted to find out the location of this gene in the mice. So when this genetic mapping was concluded, it was confirmed that the location of this gene was on the chromosome number 2. And it was also noted that this gene has the same location in the humans too. So it was concluded that many of the sequences of the mouse genome is also present in the same sequence in the human genome. So we can say that if we cut the human genome into 300 pieces and rearrange them, it will become the mouse genome, or vice versa. So the gene order is same, but there are some, using the cutting and pasting, we can rearrange the genome and we can come up with the same order in the human and the mouse. So we can say that the order of human and mice in the form of genes can be traced back in the common ancestor mammalian genome. So it means that the mammalian genome has the same orders that is present within the human and the mice. For example, we can see that the chromosome number 2 in the humans is also the same, has the same set of genes that are present in the DNA of the chromosome number 1, 2, 3, 5, 6, 7, 10, 11, 14 and 17 within the mice genome. So we can see that the rearrangement of the genome, rearrangement of the genes within the genome can result in the alteration of the genome architecture. So we cannot say with confidence that how many times this change in the genome occurs in the evolutionary history. But we can assume that a total of 250 genomic arrangements can be found in the human genome as compared to mice genome. And we can say that this rearrangement can result in the loss of the genomic structure. And we can see as example here that if we assume that this is the different large pieces within the mouse genome. This is one piece, and we cut into the large pieces, and each large piece is called as synteny. So this is synteny 1, synteny 2, 3, and so on. So we can see the order of the mice genome. Synteny 3 is placed at the first place, then 5, 2, 4, and 1. And below is the human X chromosome. This is the mouse X chromosome, this is the human X chromosome. And we can see it is in the order 1, 2, 3, 4, and 5. And the second thing to note is that the syntenies, that is the synteny 1, 2, they are the same within the mouse and the human genome. We can see here the synteny number 1 in the human is present also in the mice. The synteny number 2 is also present in the mice at position number 3. The third synteny in the human is present at the first place. Fourth is the same, and fifth is placed at the second place. So it's just a rearrangement of the syntenies within the mouse and the human genome. And the simple thing is that we can just replace these syntenies to order the sequence of mouse and the humans. For example, we can see here, if we replace this one with this one and this one with this one, number 5 with 5, 2 with 2, and we will come up with the same order of the human and the mouse. So we have to do 3 times this replacement or flipping, and we have to flip this one also with this one, and the third is this and this. And if we perform these 3 flippings of the syntenies, we will come up with the same order of the genes within the mouse and the humans. But we cannot say with confidence that only the 3 flippings can reorder the sequence of the genes within the genomes of these mouse and humans. But we can say that by using a different algorithms, especially the brute force algorithm will not work, because and search and bound algorithm will also not work here, because we have to use the greedy algorithms to find these types of flippings. And for calculating these types of flippings, we will use 2 different types of greedy algorithms, and we will calculate how many times if we flip, then the arrangement of the human and mouse genome or any other genome will be in the order.